Well, hello again. Welcome back to this part of the webinar. Sorry for this small delay. We were dealing with some IT issues on this side, but we are now able to proceed to strategic area number 6, dealing with research and development of renewable bio-based chemicals, materials, and bioenergy sources. The speaker that will talk about this area is Professor Craig Folds. Professor Folds is a professor at the Marseille University and a researcher at MIRRI. Please, Professor Folds, the floor is yours. Thank you.
Thank you very much, Luis, and thank you for the invitation to talk about this this area. Let's just go to the screen. Okay, hopefully you can see all this. So I've been asked to talk about Strategic Area 6, the research development of renewable bio-based chemicals, materials, and bioenergy sources. From the research document, you can see how this research area connects with the UN Sustainable Development Goals and the European Union calls now and coming up, and also the other research infrastructures. And I'll let you read that at your leisure. In summarizing a little bit between the alignment of the Strategic Area 6 and the Global European Area, we've written in the document that Area 6 is to develop the next generation of green, environmentally friendly and sustainable tools required to more efficiently replace fossil resource-derived materials, and more efficient microbes from natural diverse pantry, either as whole cells or from a better understanding of their metabolic process, understanding more about the function and applicability of their individual and collective enzymes as both degradative and synthetic catalysts, and finally to design synthetic microbes where natural ones are not so efficient. There are a number of relevant parts of the work process program relevant to this as part of the Europe's climate and energy targets and the European Green Deal, which are all quite relevant to this area. When we talk about renewable area, we're really talking much of the research involving culture collections focused on biofuel. But rather than going directly into this area, just some questions for us to think about now or at the end of the seminar is, will today's biomass still be the focus in 20 to 30 years? Are agro-industrial practices going to influence what we grow? Is climate change going to influence what we grow? And therefore, looking at microbes to break down the biomass now might change very much in the next few years. And especially as crops are developed to be more tolerant to climatic variations or for changing food practices, we have to take this into consideration. And we also have to consider crops at the regional continental level, where a lot of cultures and enzymes have been developed for particular crops. For example, America targeting maize, in Brazil for sugarcane. Where is Europe? Are we focusing still on wheat, or are we going to change this to high energy crops? We should also consider strains and enzymes to clean up filters and sensors in some of these other areas for the future. This is an example of a crude oil-based refinery approach where you take the crude oil and you fractionate it all down to get your high-value compounds. And this is what we're looking at in terms of biomass, where we're actually taking this raw material, the starch, the hemicellulose, the cellulose, lignin, oils, and proteins from this renewable source, and fractionating it all down into building blocks, secondary chemicals, intermediates for a whole lot of different uses and products to try and either replace directly what is currently derived from fossil-derived fuels or similar products that can be used in industry. A lot of the thing we could think about is how we use the waste material from agro-industrial practices, how we can use this area as opposed to challenging our food supply in the future. And we can get a whole range of different materials from these waste streams. We can also think about understanding metabolic processes, both in the natural sources and in the synthetic organisms, to try and understand how we can direct the metabolism to get the compounds that we are going to be using. But we still have a much further route to go to understanding basic metabolitic pathways, especially in the fungi. And we have to consider what tools and chassis are still going to be needed to really effectively turn renewable biomass into an alternative to fossil fuels. Another thing to consider is biohydrogen production. This is being developed from bacteria and also from algae. But one of the problems is we still need to break down the biomass to get into the simple sugars that can be fermented. And here we have to think about other approaches, other cultures that can do this. And one of the best ones to look at that is the fungal kingdom. And this graph just shows phylogenetic separation of the enzymes from these different fungus and how fungi have developed a very good arsenal of enzymes capable of breaking down most biomasses. What we have to do is try and use our culture collection knowledges to try and harness and understand some of these enzymes and how they work, to be inspired by the fungi in nature. We can look at the phylogenetic genomics because of the number of newly sequenced genomes coming from fungi. We can start to understand where the good fungi are coming from, where are the specific ones for biomass degradation. This is an example of a tree put together in collaboration with a lot of collaborators as part of a PhD study looking at the polyporous collection of fungi. We can use this information in growing the fungi up on different things to try and supplement industrial enzymes that are currently available. This allows us to boost saccharification reactions and try and help to identify novel enzymes which are involved in this boosting mechanism. So we can use this to, again, to cluster the fungi according to their species and to indicate what different strategies are actually being used by the fungi to degrade the cell wall. The similar thing can be done with lignin, where we can look at the evolution of different peroxidases of biotech interest to try and target lignin waste. And I think our next speaker, Ronald, will elaborate a lot more on this. But again, we can try and identify good species, and we can also try and identify what fungi are selective degraders and which are simultaneous degraders of biomass. And this will influence what we're actually looking for. If we're looking for a total breakdown of the biomass into single units, or we're looking for selective stripping of, say, the lignin, exposing the polysaccharides for further exploitation. But there are bottlenecks in industrial conversions of biomass. We know that microbes, and especially fungi, are very efficient degraders of biomass, working together to break things down in the soil. But other bottlenecks through time, we're looking at the natural degradative process compared to an industrial process where we're wanting things very quickly sped up. Are we looking only at isolated strains from our collections, or should we be looking much more on microbial consortia, either natural or synthetic, as occurring in nature? And now should we look at the different cascade systems of degradation involving enzymes and or microbes, because we can look at combinations of everything else. Rather than thinking of a one-pot system, can we actually design a much better fractionation process? One of the things we have to develop is a wider screening base. We have a lot of information on bacteria and some fungi in utilizing biomass, but we have to look at different sources as here. So here we have a list of important building blocks that can be derived from either yeast, fungi, bacteria, or chemical processes. But we can use natural strains to perform these conversions. We can engineer some strains to make conversions better or eliminate inhibitors or toxins. We can also be bio-inspired by looking at metaomics technology or co-culturing approaches, where we're not looking just at a single strain, but trying to find better combinations based on what we have available as tools within our culture collections. There's also the development of algal culture in this field, and we heard this talk this morning about the collection in Spain on algae. Or do we approach a synbio approach where we're disregarding the actual natural way and actually trying to use what we know from the genomics to create a new organism? This is an example of an algal-based biorefinery that has been published. So we know that algae can be good producers of different materials for industrial processes. We should also think about termite gut microbiota. This is a new emerging area looking at the breakdown of lignocellulose, but it's an anaerobic process and so has the issues with whole lignin degradation because of the lack of oxygen. But it is a very good utilizer of polysaccharides, the cellulose and the hemicellulose. And we also have to consider, when looking at the termite gut, is the development of enrichment techniques to try and really stimulate what is targeting the hemicellulose or the lignocellulose. We also need to develop a wider panel of materials to screen. It's no use having nice culture collections if we are unable to actually screen them for industrial applications. This is an example of a paper that we have just published in Marseille using the serum CF collection. So the serum CF collection is a filamentous fungi, mainly isolated from lignocellulose, from woody material, rather than food-approved organisms. But we can look at things like textile dyes for wastewater treatments. We can look at lignosulfonate, an example of lignin coming from the pulp and paper industry. We can look at improneal as an example of a plastic type material. And we can look at cellulose as well and how we can break things down into glucose. So we can use our culture collection on this at the moment to screen for useful products and building blocks for industry, but we need to do more. We need to know what substrates can we use in plate assays or in deep well assays, and how we can detect changes in an easy way using these compounds. From results of screening like this, we can identify groups of fungi which are very good at all degradation or in one particular type of degradation, such as industrial dyes or bioplastics or lignins. And so we can use this information coupled with the genomics to really identify good strains for collaboration with industry. The second part of the strategic area also talks about climate change and how this is going to have an impact on the industrialization and use of cultures. The document says with urgent need for renewable energy to tackle climate change. And this came up just at the weekend here in the Glasgow Climate Pact, where research on biodiversity were really highlighted in the document that came out from the COP26 meeting. We know the planet's heating up. There are changes. There's going to be changes over the next number of years as the temperature rises, and this is going to have a big impact on what we can grow, what we can utilize, and also the natural biodiversity existing in both the soils and the plants, and what we can harness in this area. This is just an example you can see in France, there are certain areas which are really going to be heating up over the next few years. And this is where culture collections can come in, and we can use these culture collections, especially if we have quite a global culture collection at our availability, either in one partner or across the partnership in MIRRI. In Marseille, the fungal collections have come across from all different regions of the planet, and we have this is pre-Nagoya, so it was a little bit easier to collect the cultures. But we have at our disposal different fungi representing a whole lot of different mechanisms and growth capabilities. We can start to look at, say, even a single type of species like Pycnoporus or Basidiomyces, collected from different parts around the world, and we can compare this on their ability. Some of these cultures will be adapted to be growing at higher temperature or under environmental challenge conditions, and we can use this natural knowledge to harness our development of industrial tools. For example, this was again with the Pycnoporus species. We looked at functional lacase diversity from the different areas from here, and we found that even though these Pycnoporus strains can produce lacase, their activities, their basic lacase activities do vary depending on the strain and the location that the fungal strain was collected from. So we can use this information in developing our processes. We also should look at more at extremophiles. We have talked about this this morning especially, but looking also at extremophiles and alternative solvents, how cells grow, how enzymes work in non-aqueous solutions like organic co-solvents, where some of the work I've shown, you have an increase in activity when you start to replace some of the water with solvents. The same with ionic liquids. Although we have an inhibition of cellulases, we can get induction of higher activity in other enzymes. So we have to look at what concentration of ionic liquids an enzyme or a microbial cell can actually work in. And another area is deep eutectic solvents. The idea here is potable water in the future is going to be quite limited. So can we design industrial processes to work in non-potable water, either in high salts or in presence of co-solvents? And one of the examples is the marine microbes, which are very tolerant to salt levels and polluting material in the water. At the moment, these organisms tend to be studied for secondary metabolite production with respect to new drug discoveries, but we have shown that some enzymes are produced under salt stress conditions, and these enzymes are tolerant to the presence of salt. So here we are adapting our enzymes, adapting our knowledge and our culture collection to foreseeing what industry might need in the future. This is just an example of some of the novel salt.
Sorry, Craig, to interrupt you. We have to speed up a little bit, please.
Yeah, okay, I'm getting to the end. So we have salt adaptive enzymes coming from things like the mangroves, where we can compare what is induced by salt, what is not induced by salt. We also have to design mimicking climate change facilities in the laboratory, looking at how we can study salt effects, how we can look at changes in temperature and in changes in pH, all of which will be influenced by climate change. And for this, we need multiple partners, not just culture collections, but genome sequencing, annotation of the enzymes involved, industrial uses, and industrial collaborations. We can't just do it ourselves. We need collaboration. Potential future trends, we just sort of brainstormed the other day. We have to look at exploring co- and mixed organisms and consortia. We need to develop better panels of substrates and screening methods, looking at catalysis and non-aqueous solvents and non-potable water, looking at how we can turn the cells and enzymes into biobatteries, biocomposites, material from fungal mycelia themselves, activated soil enhancement we've heard about just this afternoon, and also biocapture of rare earth metals, minerals, etc. can all be done by our organisms in our culture collection. And also finally looking at the deconstruction of mixed waste from vehicles, electric car batteries, municipal and commercial wastes. So these are things for the future, and I thank you for your attention this afternoon. Thank you.
Thank you. Thank you so much, Craig, for this great presentation. Your last slide was actually seeming to be a full list of topics for a future event that MIRRI can hold. Very interesting, very, very actual indeed. We will be staying on this topic with the case presentation entitled Bioconversion of Lignin-Derived Aromatic Compounds into Platform Chemicals. The speaker is Professor Ronald de Vries. Professor de Vries is a professor at the Utrecht University and also a group leader at the Westerdijk Fungal Biodiversity Institute. Please, the floor is yours.
Thank you very much, and thank you for inviting me to give the talk here. I think indeed, as Greg already mentioned, I will follow up a little bit on his narrative. Actually starting a bit that indeed, of course, our culture collections provide us with lots of options, but as Greg already mentioned, without understanding what we should screen for, we can't really screen our culture collections to the optimal benefit. So what I'm going to talk to you much more about here is not so much the diversity, but to understand the process that we can then use as a potential option to see which fungi would be most suitable. So as mentioned, I will talk to you about conversion of lignin-derived aromatic compounds. And first thing I would like to tell you, so if you want to establish cell factories, and especially in this case aromatic cell factory, there's a number of things you need to take into account. First of all, if we're looking at lignin-derived aromatics, we're not talking about a single compound. We're talking about a range of different compounds, because typically, whichever way you degrade lignin, you get a pool of aromatic mono and oligomers. So if you want to use these pools of compounds, you really need to try and find a way to convert them into platform chemicals or single compounds, or at least simpler mixtures, so that they can be used to produce a whole range of different products. So to be able to generate a cell factory, there's a couple of things you need to have. You need to first of course, understand aromatic metabolic pathways very well. You need to have tolerance of the organism to aromatic compounds, and as many of you will probably know, aromatic compounds tend to be quite inhibitory to most microorganisms. Of course, the cell factory needs efficient genetic modification methodology like CRISPR-Cas9. And of course, it would also be very advantageous if you already have good fermentation properties, as this will really help in the downstream upscaling of the process. So unfortunately, when you're talking about aromatic metabolism, we actually didn't know that much up to a few years ago. I mean, there were a lot of papers out, especially in the 70s and 80s of last century, where basically people had been measuring conversions of aromatics, and we've summarized them in the figure you see here. So basically, most of the papers would say something like, okay, if I grow on conophyryl aldehyde, I can see that the fungus makes conophyryl alcohol. Or if I grow, for instance, on vanillyl alcohol, we can get vanillin. But how this happened, which enzymes were involved, which genes were involved, that was largely still a black box. So we started the project, which I will talk about today, by first categorizing all the literature that was there and putting this into a single scheme. Now, I have to make a little comment here. This scheme looks very nice and very intricate, but this is based on studies in many different organisms. So there's absolutely no guarantee that a single species would actually have this full capability. So a PhD student of mine who graduated this year, he basically then continued on that, and he started putting these things in a bit more applied context, starting from the lignin building blocks and mapping the pathways towards the ultimate ring cleavage, which is the main aim of most microorganisms for aromatic compounds, because once you do ring cleavage, you lose toxicity. Now, what you can see here very clearly is that there are substantial differences between fungi, bacteria, and even yeasts, because as you can see by the color boxes or color buttons which are underneath, not all steps are shared between all these organisms. Now, we, of course, were focusing on fungi, and for this particular project, we took Aspergillus niger as our reference organism to see if we could make an aromatic cell factory. Now, the reason we picked Aspergillus niger, of course, that it has a long history as being a fungal cell factory, particularly for enzymes and the production of citric acid. It already has those good fermentation characteristics, which I talked about earlier. It has also very efficient genetic methodology, including CRISPR-Cas9 genome editing. Also, of course, it's nice that many products from this fungus have already achieved GRAS status, so that creates a certain history of use. And also, very important, it's one of the few fungi that has a gold standard genome sequence. And for those of you not so familiar with genome sequencing, a gold standard genome sequence means that the genome has no gaps, so all the gaps have been filled by additional sequencing to have one continuous sequence for every chromosome, and on top of that, the annotations have been manually curated. And there's only about 2 or 3 fungi that currently have that. So I'm going to give you a few examples. I mean, this was, to be honest, this was a very productive PhD student who has PhD so far resulted in 10 papers. I'm just going to give you a few examples of his work to show you what kind of things we found and what this may actually mean. So one of the things we found is that you can actually produce a compound called cis-muconic acid by a relatively simple metabolism in fungi. Now you might wonder, so why is this relevant? Well, just to put it here into context, if you go to Sigma Aldrich and you buy salicylic acid, you pay about 62 euros per kilogram. But if you want to buy cis-muconic acid, you pay more than 29,000 euros per kilogram. So just by doing this simple conversion, you have a 470-fold increase in value. At the moment, cis-muconic acid is mostly produced chemically from fossil resources. So also being able to produce this from more renewable resources would be highly interesting for industry. So cis-muconic acid you find in many different things like polyurethane, in resins, in nylon, in pads, and in lubricants. So it's a very commonly used compound in industry, and the estimated market in 2017 was about $54 million. So what we did in this case, we tried to find the enzymes that were here in the question marks. This one was already known. We also didn't know this one. And we did this by a combination of transcriptome data and literature, where we also looked at different opportunities with respect to PFAM domains and other aspects. So based on this, we found 6 candidate genes, which are listed down here. And we basically made mutants in those genes to see what would happen with the different intermediates of the pathway if you grow these mutants on it. And what you can see very quickly is if you delete this gene, we see reduction of growth on salicylic acid. If we delete this gene, which was already known, we see reduction of growth in 2,3-dihydroxybenzoic acid. And if we delete this particular gene, we see that it's reduced on both of them as well as on catechol. So based on this, we can of course place these genes in the pathway, and we can basically see that SHIA is sitting here, whereas the CRCA is sitting at this position. We had various screenings for that. Greg talked a little bit about this as well. Plate screens are of course very important to quickly look for these activities, but I will not go into detail into that today. So we then wanted to have a look. So we now found the genes that were responsible. So if we produce these enzymes, can we actually do a 2-step conversion of salicylic acid into cis-muconic acid? So what we did is that we produced these enzymes in Escherichia coli. We produced them as initially as crude enzyme extracts, and we basically had a look what would happen if we provide these incubations with these enzymes with the different components. So if we, in here in the left-hand side, you see the example that if we have no extract or if we give the empty vector control, nothing happens to the salicylic acid, we only have salicylic acid. Whereas if we also add SHIA, we can see that part of the salicylic acid, and it depends a bit on the transformant we used, is converted into catechol, suggesting that at least this first step we can do with the enzyme we identified. And then we had a couple of enzymes by themselves, which are supposed to be responsible for the second step. And you can see if we just use that enzyme, we don't make catechol from salicylic acid, so we also don't make cis-muconic acid. But if we combine some of these things we can still get to catechol. But the interesting thing happens, and I think something just went wrong in that slide. Okay, sorry. Okay, so for some reason part of the slide fell away. But I can already tell you now that if basically we also add the CRCA, so if we combine these two enzymes and we give this to a mixture with salicylic acid, basically we do not have any catechol or salicylic acid left at the end. We have almost 100% conversion into cis-cis-mucomic acid. So by simply identifying the genes in the pathway, we found a very simple process of doing this conversion, which could be industrially very interesting. Now, in light of time, I will not give you too much more details on that. The results have all been published, so you can find them in the publications. So I want to give to you the second example here, which is the production of protocatechoate. Now, this is a very interesting compound because in the pathway, it's more or less a central compound where a lot of other compounds end up before ring cleavage takes place. In addition, industrially, it's quite interesting because protocatechoate is used as a building block for polymers and plastics, but also in several pharmaceutical applications. So if we can do biosynthesis of protocatechoate, we have a very high potential. So again, we try to find the genes for this pathway, and it's summarized here. So this is basically the part of the metabolic pathway that ultimately goes to protocatechoate. And what you can see is that many things can end up here. So we see caffeic acid going there, pyrocomeric acid indirectly, just like cinnamic acid, and also many of the other compounds in the bottom part of this figure. So the idea we had is that if we can identify the genes that go towards it, which are these ones, BPHA and PHHA, and we can identify the genes that result in ring cleavage of protocatechoic acid, we should be able to accumulate this compound. So we used the same approach as I mentioned for the previous one. We used transcriptome data together with genome analysis and PFAM domains and other features, and we tried to identify the candidate genes. And you can see that we were quite successful here. So basically, we don't see a phenotype for this gene, which is one of the two options for indirect ring cleavage for protocatechoic acid. And the reason, of course, for that is that there is still an alternative option. We found PRCA, which basically goes here, and there for some of these compounds, we have a strong phenotype, suggesting that this is the preferred pathway for protocatechoic acid conversion. Then we also made some double mutants, so we have the double mutant here for VA PRCA, so that's this double mutant, and you can see that on most of the compounds now, we do not see any growth, with the exception of phenylic acid and ferulic acid. And that's basically because these two compounds do not follow this pathway, but go a different direction. And we've published that in a later paper as well. So this suggests that with clever enzyme engineering or genetic engineering, we can actually not only get the enzymes that are required to make protocatechoic acid, but theoretically, we should be able to generate a strain that doesn't convert protocatechoic acid and accumulates it from many different aromatic compounds. So that's, of course, what we tested next. So we basically made this strain, which has the double deletion, which cannot convert protocatechoic acid anymore. And we saw how much protocatechoic acid would we get if we supply this strain with various different aromatic compounds. And you can see here, of course, if we provide the protocatechoic acid, nothing happens. But all these other compounds, so the protocatechoic aldehyde, benzoic acid, anisic acid, pyrohydroxybenzoic acid, comeric acid, caffeic acid, all of these compounds result in a nearly 100% conversion of and production of protocatechoate. Now, there are some compounds which didn't work quite so well. So like here, we see cinnamic acid and cinnamyl alcohol that didn't work so well. And that's because for cinnamic acid, there is a second pathway which goes ultimately to styrene, which basically is an escape for the blockage we made in this strain. The other compounds, which you see down here, as I mentioned already in the previous slide, did not result in the production of any protocatechoate, which means that basically these go into a different pathway. So this was, of course, a very nice result because it shows that if we take this strain and we give it different aromatic compounds, it will actually result in the production of protocatechoic acid from all of these. So what would happen if we would give it a mixture? Does it work just as well? And this is what we did here. So we did this mixture of aromatic compounds, which you see down here, and we had 2 millimolar of 5 different aromatic compounds, And what you can see is that we produce 10 millimolar of protocatechoic acid. So this strain can actually take this mixture of aromatic compounds and convert it into a single product. But also if we take, for instance, this bitter almond oil, which is quite rich in various aromatics, we can see that we can get a very nice production of protocatechoic acid. So what we effectively have here now, we have a fungal strain that can do an almost 100% conversion into an interesting industrial product without requirement of any other chemical treatments. So with that, I would like to conclude, and I think that you will see, if you would go through all the papers which we've published in the last 3 or 4 years, that many of the aromatic pathways in A. niger have now been identified. Now, that of course looks great, and it basically gives us a really good starting point. But of course, being at a biodiversity center, we've also looked into the conservation of these pathways in other fungi. And unfortunately, what we see is that we see lots of diversity in the pathways. There are certainly commonalities between fungi, but even within a single genus, like within the Aspergilli, we can see that not all of the species have the exact same pathways, and sometimes they've basically made a different choice. So using different fungi to make aromatic cell factories, you might need to do a lot of the analysis again for the missing steps. So I think also the last slide showed you that the generation of fungal cell factories for aromatic compounds based on waste streams, especially lignin, is certainly a realistic approach and something we will explore in the future. So with that, I would just like to thank the people involved in this, and especially Ronnie Lubbers, who was the PhD student who did most of this work and was incredibly productive. But of course, he was also supported by various other people in the group. And I definitely also want to mention the Joint Genome Institute in the USA, where most of the transcriptomics was done. And with that, thank you for your attention, and questions will come later.
Well, thank you so much, Ronald, for a very interesting presentation, very nice results, and a very productive PhD work indeed. We do have time for a very quick question, if there is one. Marta?
We have one question from Mateus Wilk. Do the pathways differ between the different strains of the same species, or they are at least species specific?
I mean, we haven't tested that in detail. I would say that the pathways are likely the same, but the flux through it and the efficiency differs from strain to strain. And of course, also the tolerance for aromatics can differ from strain to strain. So you may have the same metabolism, but the ability to survive may differ from strain to strain.
Thank you very much.
I'm sorry, I hit by the mute by accident. I was saying that we can now move on to the strategic area number 7. It's about rescuing and preserving microbial biodiversity. The speaker is Gerard Werkleif. Gerard is a curator of filamentous fungi at the Westerdijk Fungal Biodiversity Institute, and also a member of MIRRI's International Coordinators Forum. Gerard, the floor is yours, please.
Thank you very much. Yes, I'm grateful for the opportunity to speak a little bit on strategic area 7. Some of the aspects that are very important here have also been touched upon, of course, earlier today. And the main theme is to rescue and preserve microbial biodiversity. So it has been mentioned before that there are important Horizon Europe missions that match very nicely with the program of MIRRI and with our strategic ambitions. Caring for soil is caring for life, and also Mission Starfish 2030, restoring ocean and our waters, are 2 examples of that. It is very clear to us that these missions can only be successful when we have a better understanding of the composition and the function of natural microbial communities. And these communities play a key role in supporting all biota, ecosystem stability, and also in securing or restoring the ecosystem services. Now, as microbial biological resources and centers, we are key players for microbial biodiversity investigation and preservation because we deliver the data and the materials needed for research. And some of the culture collections are over 100 years old in our community. And they often hold very unique materials, isolated from pristine habitats or habitats that have been seriously affected or even disappeared since the isolates were gained. So new resources are, of course, every year added to the culture collections, and MIRRI has now provided a very nice single point of access for people to browse in the microbial resources that are available for the user community. Europe is still a stronghold for ex situ conservation of living microbes. The culture collections of the partner institutes represent over 400,000 strains of microbes originating not only, of course, from Europe, but also from around the globe. And it must be said, preserving microbial biodiversity ex situ is really a global challenge. In this graph, you can see a summary of the largest public microbial BRCs in developed countries that have significant fungal holdings. This is from a paper that we wrote some time ago to highlight the fungal biological resources for the development globally. And you can see here that on the different continents, there are very large culture collections of which various parts are fungi and yeasts, and the rest are other organisms, mainly bacteria in most cases. So Europe is a stronghold, but we have to, of course, look further than that. So what will MIRRI's approach be to the preservation of biodiversity and to the endangered environments? Well, of course, we will work on to improve the ex situ conservation methods, and this is really necessary, and also come to ways of keeping environmental samples that contain more than one organism and even contain consortia. But also, it is very clear that even in our collections, there's a lot of hidden biodiversity. So we need to discover, describe these materials again. And we also have to preserve novel organisms that come from all kinds of activities within the framework of MIRRI and the partner collections, of which some examples will be given later, from arable environments, from natural environments, etc., etc. We need to characterize the strains, but we also need to study and characterize the microbiome samples at the phenotype and the genomic level. And we will collaborate with other research infrastructures to more efficiently integrate and disclose the data that we have on all these resources in our collections. And these data and this expertise that we have will be important for developing soil and water sanitation and remediation approaches, as several examples have already been shown very nicely today by my colleagues. So MIRRI will link biodiversity with bioprospecting. It has been mentioned before. Soil microbes are promising sources for new therapeutic drugs. Marine microbes are particularly interesting for applications in pharma and nutraceuticals, and also, of course, for the development of bioplastics and other biomaterials. MIRRI will explore the soil, marine and freshwater microbiomes for novel microorganisms, and also preserve them ex situ, develop methods to do that successfully. And all these resources can be used for bioprospecting and other screening activities to find new sustainable applications where microorganisms can help to support food security and health. Now, if we're thinking about rescuing microbial diversity, maybe locally present in the environment, but also we are talking about resources that have been isolated with a lot of effort before, and that have been kept in laboratories, have been worked on by many often very active researchers. And in many cases, these collections are on the threat of being lost because the organizations that have housed these important collections can no longer preserve them. Why are they important? Well, of course, there is a lot of hidden biodiversity in these kind of collections too, and a lot of undescribed biodiversity. And also these strains can originate from ecosystems that have been destroyed or are difficult to access again or against only a lot of costs. So to mention some examples that we have managed to preserve at the Westerdijk Fungal Biodiversity Institute in our CBS and NCCB collections for respectively bacteria, fungi, yeasts and bacteria are the following. The Martha Christensen soil microfungal collection that has been kept in the USA for a very long time with materials from many different ecosystems in the USA and other parts of America in the tropics, over 2,500 strains. A very important collection of mainly plant pathogenic and soil-borne hyphomycetes with dark structures, the so-called diminutaceous hyphomycetes, that were collected over a lifetime career of Dr. Emery Simmons. Many plant pathogens, over 400 strains. The very interesting and important marine yeast collection of Jack Fell that contains many deep sea resources, over 700 strains coming from around the globe as well. And then the insect-associated yeast collection of Professor Meredith Blackwell, who retired a few years ago in the USA. And we're very glad that we could preserve this very important collection with over 1,500 strains. So another example that the Westerdijk Fungal Biodiversity Institute has been involved in is that we form a roadmap consortium on the national roadmap together with other institutes, Naturalis Biodiversity Center, the University of Amsterdam and the University of Twente. And in this NIBA consortium, we have managed to successfully apply for a roadmap application that gives us the possibility to start the project ARISE. There's 14.4 million euros available from the Dutch National Roadmap funds for large-scale infrastructures. Authoritative and rapid identification system for essential biodiversity information. That is what ARISE stands for. Well, what are the ambitions of ARISE? It's a project that has an initial funding of 5 years, but the intention is to build an infrastructure that will enable us to use it in the future. And the main ambitions over the long term are to name all Dutch biodiversity species in the Netherlands, to being able to identify any sample from any location and by at least a lot of methods. Here you see the team in Westerdijk Institute that is affiliated with the project, working mainly and focusing mainly on soil-borne fungi. ARISE vision is to underpin that the biodiversity is the key for the planet and human survival, and also to capture the loss of biodiversity, which is crucial for our future. So there will be several components. There will be the identification on any level, any location, as I said, for knowledge and research. There will be a BioCloud digital service that allows us to facilitate complex biodiversity research questions and also to study species interactions at any level and to also monitor the stability and the resilience of ecosystems. So the proposed infrastructure has several components. There is the Netherlands Species Reference Database, which will be in part, of course, on fungal resources, but also on plants, animals. And this is mainly the database that allows us to harbor all the data on validated species. There's a sequencing facility that is distributed over the Naturalis and also the fungal and our institute. There is the species identification engine, which will mainly consist of algorithms and apps that allow us to rapidly identify the higher organisms. There's, of course, the biobanks. There's the living collections of the fungi and bacteria in the Netherlands that are all centered now in the Westerdijk Institute. The BioCloud, as I told you, for enabling research facilities to combine data and interlink data. And then there's demonstration sites for the monitoring. There are a few sites that have been selected in the Netherlands, both in urban as well as in natural environments. And There will be a continuous monitoring of biodiversity at any level, and also regularly samples taken for use in the isolation of fungi, the fungal components, but also to do entire DNA extractions, eDNA extractions that will be used for metabarcoding activities. So ARISE will produce a lot of results, and many of these results will be very relevant for MIRRI. In our CBS collection of fungi, we have over 8,000 isolates from soil. A lot come from the Netherlands, but there's also quite a number of isolates that originate from surrounding countries, and these will all be taken into account into trying to characterize the soil fungi to a level that we can better understand the diversity that is present in the soils in the Netherlands, both in pristine soils as well, as far as they are there, as well as, of course, all human influence soils. And we do a special emphasis then on comparing the soil isolation results with the metabarcoding results that will be generated by the team of Naturalis. Then to conclude, there is a project on which we are momentarily very active that targets the genus Fusarium. Fusarium is a genus of ascomycetes that are notorious for the fact that they are plant pathogens, but also are emerging human and animal pathogens. They're also producers of several very important mycotoxins like the fuminosins and the trichotizins. And although the genus has been studied for decades, well, more than a century, in fact, mainly, of course, for a long period by the morphological and phenotypic analysis of the asexual forms of this group, it is very clear that by implementing the molecular methods, we have discovered how hugely diverse this group is, and that it is really very important to understand all the individual species complexes that are present in this group of the Nectiaceae. And so together with other collections, such as the IMI collection in the UK and several other collections, Professor Pedro Kraus is very active now in starting this project and further characterizing the species complexes that have not been very worked on in depth, look at morphology, biochemistry, phylogenetics, and more. And this kind of information and also the characterization of the fungal isolates that are in any of the collections, including the MIRRI collections, will, I think, considerably further our understanding of this very important group. And this is just an example with which I would like to conclude this contribution. Thank you very much.
Thank you very much, Gerard, for this presentation. It's actually a very important, a critical area for MIRRI, of course, because it does make the foundations for the rest of the areas inside MIRRI. It will only make sense if this exists. So we can move to the case presentation now. It's about preserving yeast diversity from worldwide endangered cold habitats in the era of climate change scenarios and challenges. The speaker is Professor Benedetta Turchetti. Professor Turchetti is a curator of DBVPG, the Industrial Yeast Collection at University of Perugia. Please, Benedetta, go ahead. The floor is yours. Thank you.
Okay, good afternoon or good morning, everybody, and thank you for giving me this possibility to talk during this webinar. So, as everybody knows, microorganisms are the pillars of life on Earth, and over a billion of years, they have evolved into every possible niche on the planet. But only in the last decades, we have started to peer deeply into the microbial cosmos. And what we have found is that microbial diversity is represent a genetic and enormous genetic and biological pool, and that can be studied and also exploited for their new products, new and particular, peculiar metabolic pathways and metabolism. and also for, why not, novel genes. But the principal obstacle in such direction is that this enormous diversity is largely unexplored. And if we want to compare microbial diversity to figures, we can compare to an iceberg, for which only the upper part of the iceberg represents the already known microbial species, the ones that we have characterized and we know, and the hidden part, the lower part, represents the diversity that should still be described, and so the unknown microbial diversity. And this is due mainly to two main reasons. If we, for example, look at the number of estimated fungal species, this range between 2.2 to 2.8 million. But the number of the species that have been already described is estimated is estimated to be around 120,000 species. And this difference is due to two main reasons. The first reason is that there are a lot, as Gerard just said, there are a lot of different habitats, environments that are completely unexplored, because probably too difficult to reach, or extreme habitats that are completely unexplored. And the second main issue is that a serious evaluation carried out in different type of environments show that only a small percentage, and for fungal species is around 11-12% of the species present in the environment are culturable in laboratory in synthetic media. And this is what we have done at the DBVPG collection. The collection started the exploration since 1920s, and in those years the studies were mainly focused on fermented beverages, and they were able to isolate something like more than 1,000 strains of Saccharomyces cerevisiae and related strains. Then the study at the collection moved to the natural environments, tropical habitats, and in the last 20 years, we focus our attention on glacial habitats. And now we reach around more than 7,000 strains conserved in the collection. And why glacial habitats? Why in the last years we focus our attention and we decided to investigate glacial habitats? As everybody knows, because the Glasgow Climate Pact of last week, we have a very enormous problem, that is the climate change, that is due to greenhouse gases emission is creating a global warming. And the global temperature is rising in the last year, and it will rise in the future. And more notable enhancement of temperature is expected on terrestrial region than over the ocean. And this will result in the modification or even the loss of many habitats. And among these habitats that are going to be modified or even lost, Arctic, Antarctic, and high mountains glaciers are regarded as climate change hotspots, because the presence of a psychrophilic biological diversity that are present in those habitats and that were evolved and adapted to the cold, and therefore are extremely vulnerable to the increase of the temperature. So this habitat and the biodiversity that they represent are going to disappear. So this is one of the main reasons why we focus our attention on glacial habitats, because they represent an endangered environment characterized by a microflora that is disappearing with it. Then, of course, there are other reasons. These habitats are considered among the most undersampled habitats. So there are very few ecological studies related to the distribution of microorganisms and especially of yeast in cold environments. Then these extreme environments are extreme environments, and so the presence of human is scarce. And this means that we can study the ecology without any activity of human on those habitats, so in the real natural ecology. And last but not least, the microorganisms that colonize cold environments developed year by year particular and peculiar survival strategies, and so specific metabolism, the possibility to produce specific molecules that can be very useful for biotech purposes. So I want only to show some pictures that are very from where we can see what's happening at glacial habitats. Here we can see that in less than 80 years in Alaska, we can see what happens. So a very big glacier is completely disappeared. And this is what happened in Alaska, but also, for example, in Himalaya, at the Baltoro glaciers in the Karakorum, that is one of the highest mountains in the world, and also in this case in the Caucasus in Georgia, where a glacier is completely disappeared and we have only trees. So this is what is happening and is going to happen. We are losing environments, cold environments, and we are losing also the biodiversity conserved on that. So what we have done in the last 20 years, we are organizing different sampling campaigns in Italy and especially in the Alps, but also in small glaciers in the Apennine, in the center of Italy. And then we also explore Antarctica, and in particular Victoria Land in this Antarctica. And what we have done is to sample different kind of materials, so snow, melting water, ice core, super and subglacial sediments, but also permafrost, ice cores, brines, and rocks. And so we do what in the collection we used to do. So we isolate as much as possible his diversity. So from water, we use filtration, and then we go for solid sample with sometimes also with enrichment protocols. And of course, after isolation, we identify all the diversity using molecular methods. And then we conserve everything inside our collection in lyophilized and freeze form. Well, what we have found, we were able to isolate more than 1,000 strains belonging to 78 species. And these species were almost belonging to the Basidiomycota phylum. Only a few were from Ascomycota, and in Basidiomycota there are a large prevalence of the subphylum Puccineomycotina. And the very interesting thing is that the 20% of the isolated species were new species, so species never described before. And this 20% of the isolated species represents more than 12% of the total strains that we have isolated. So the species with characteristics such as to differentiate them from the known biodiversity. So they represent a novel characteristic and features. So the question is this reason sufficient to continue to isolate and preserve, especially in environments that are peculiar and that are disappearing? Of course, our answer is yes. So I want only to show some of the new species that we described. We described a new genus, the Glaciozyma, with the two species, Martini and Watsoni, or both are unable to grow at 20 degrees. Then we found a lot of species, a lot of strains in Brachia genus, and we described Brachia roberti, Blolopis, montana, fibulata, and stelvica. And then we found also some Schistobasidium, in particular alpinum, and of course isolated from Alps, and then Gustinia terre, that was represent again a microorganism unable to grow at 20 degrees. And then 3 new species in Naganiscia genus, Onofri, Vogam martini, and Nivalis, and other species like Enterocephalacria gelida, Piscorozima silvicultrix. One species that is in common with the habitat, the cold habitat in Patagonia, in Argentina, that is the Diossegia patagonica. And then one species that is the only one in Ascomycota, that is Staphrina antarctica. So, low or even sub-zero temperature influenced the life of all organisms in multiple ways, contributing to the development of harsh environmental conditions. So not only low temperature, but also low activity water, low organic nutrients availability, high hydrostatic pressure, high solar radiation sometimes. And all these characteristics all this stress, the microorganism react with different adaptation strategies. So, for example, they change the chemical composition of cellular membranes, or they started to synthesize protecting proteins or cryoprotective macromolecules, or they started to reduce the growth rate, or they starting to synthesize cold active enzymes, or intracellular, or starting to accumulate intracellular lipids. And of course, obviously, we try to exploit this characteristic, moving from the taxonomy to taxonomic study to applied research through continuous screening of these strains. For example, all the strains isolated were repeatedly screened for the production of extracellular cold hydrolytic enzymes. And what we have found, for example, is that a large majority of these strains were able to produce extracellular protease and lipase. And these enzymes were much more abundant when the strains were grown at low temperature than high temperature. And if we compare, for example, so we compared the enzymes production of cold adapted yeast and yeast isolated from tropical habitats, and we would be able to show that the abundance of the enzymes for the tropical habitat isolated were more present at 20 degrees, while the enzymes produced by cold adapted strains were much more present at 10 or 4 degrees. And so the isolation habitat affects the activity of the product or the produced enzymes. And this is a very interesting information, not only for explaining the ecological role of these strains in the cold environment, but also for a possible use of these enzymes, for example, as detergent additives for eco-friendly cold water washing, or for example, for enzymes for food industry, where this characteristic is very appreciated. Then we also look at the aptitude of these strains in the aptitude in the accumulation of lipids. And in particular, we also in this case we try to compare the lipid accumulation of psychrophilic and mesophilic strains. And what we have found is that the psychrophilic strains were able to accumulate much more lipids than the mesophilic one. So here you can see that there are a lot of much more cold adapted yeast were able to accumulate much more lipids. And also another interesting thing is that the cold adapted yeast increase the unsaturation of the fatty acids that compose these lipids. And we have a large majority of oleic acids among the fatty acids of the lipid accumulated. A few strains were also selected for their ability to use different carbon sources, and in particular, we focus our attention on lignocellulosic biomass that, after a physical and hydrolytic activity, were used as substrate for the selective oleaginous yeast for the production of lipids. And these lipids were also extracted, and the fatty acids that they produced were quantified, and in order to use them for different purposes. First of all, for example, for biodiesel production, but also as building blocks for lubricants, for adhesives, solvents, biosurfactants, cosmetic, and also biodegradable polymers, bioplastic. That is very, very interesting topic in this period. Then, moving from the industrial to food and beverage application, we wanted to explore the peculiar characteristic of the strains in the genus Mrachia. Mrachia, as I said before, is a genus that is well represented in the cold-adapted environment. And in our case, we want also to underline that Mrachia is very interesting because it is one of the few Basidiomycetes genus that is able to ferment. And so we want to look at this ability, and we try to use Mrachia for the production the production of beer. So starting from malt. And why beer? Well, beer market is always looking at new insight for consumer, and this insight is almost focused on low calories beer, low alcohol or free alcohol beers, and functional beer, gluten-free beers, and new flavor beers. And the use of non-Saccharomyces species to beer production. Can you still see the presentation? Okay. Okay, so as I said, so the use of non-Saccharomyces species for beer production is a natural way to introduce in the market new products. And so we try with Mrachia. We screen different strain and different species of Mrachia, and we were able to select one strain of Mrachia gelida, and we compare the ability of Mrachia gelida to produce new beer to the commercial starter that is Saccharomyces lutefisk. And what we have found is that we were able to obtain a beer with low alcohol content, and this is what we want because the market asks for low alcohol content beer, and so also low diacetyl production, so more or less a good beer. And additionally, the organoleptic characteristic of the beer obtained using Mrachia gelida were much more appreciated by the panelists than the commercial starter. So probably this will be a new starter, a possible new starter for the future. So concluding, these are only some example of what cold yeast potentialities, of the potentiality of cold yeast that, thanks to the sampling campaigns in cold habitats, we are able to isolate, and we are still conserving in our collection. The collection represents a treasure chest and of yeast in our these are also, especially in our ways, they are in danger of disappearing, and that's this treasure can be exploited now, but also in the future. So don't forget what we have in our collection. I want to thank you all our partners and the different projects, and thank you for your attention.
Thank you so much, Benedetto, for this presentation. I think it was a great way to illustrate one of MIRRI's mottos, from microbial collections to real-life innovations. And personally, I have to add, beer is one of my real-life innovation favorites. So it was a very good way to conclude this strategic area. So we have time for a quick question, if there is one. Apparently not. Marta?
I don't see any questions in the chat, but I could personally ask one if Benedetta agrees. It was a fictional one. If you have to sample a new environment, which one would you choose?
A new one from, well, my dream is to samples in tropical forest. or you know, yeah, in some tropical forest is my dream.
I wish you to do it.
Thank you so much. Thank you so much.
Okay, thank you. We can now proceed to the last presentation of this webinar. Many of us, of course, are looking forward to hear from Marta Conde Vidal. Marta is the national contact point in Spain for the cluster of food, bioeconomy, natural resources, agriculture, and environment. So this is a very challenging task, I imagine, to present the opportunities inside this cluster because it has such a broad and a wide spectrum. But I'm pretty sure that Marta will enlighten us about these opportunities. So please, the floor is yours. Thank you.
Thank you very much, Luis. Good afternoon to all the people who are listening. It's for me a great pleasure to share with you, to bring you all the opportunities that you have already opened within Cluster 6 calls in Horizon Europe. I will share now my screen. Okay, I think you probably see now my presentation.
Indeed, you can see it. Thank you.
Okay, as Luis said, I am Marta Conde. I am one of the national contact points in Spain, supporting applicants within this broad cluster, including research and innovation in food, bioeconomy, natural resources, agriculture, and environment. As you probably know, Horizon Europe is the new research and innovation program for the next 7 years. And around 10% of the budget, near 9,000 million euro, will be dedicated to support research and innovation in these specific fields. I am very happy to hear the previous presentations because it's a pleasure for me to have such an interesting audience listening and with the ears open to all the opportunities that this program has, because indeed all the scientific areas that I've heard meet or are absolutely linked to the specific challenges included in this cluster. But I am sure you are aware that making research at European level means to be in line with the European politics. And within this cluster, you know that it's very broad. You have to be able to keep a solution and reach the objectives that are included in many different policies. In the one hand, the agricultural policy through the EIP Agri, the bioeconomy strategy, the several strategies and actions plans that are being implemented within the European Green Deal, and some others like the integrated maritime policy and so on. So you're all focused very much on your research, but please don't miss or don't forget to link your research to the political priorities from the European Commission. And particularly in this moment, as you know, the European Green Deal is the focus of the new European Commission, and its main objective to produce this change in Europe so that we are the first climate neutral continent, Europe, by 2050. And according to this policy, to this European Green Deal, as you know, many strategies and action plans are being implemented, linked to the calls that you will see in Cluster 6, as I will explain now. And as I said some minutes ago, we have near 9,000 million for the next 7 years within this Cluster 6, but we have to take into consideration that around 10%, around 1,000 million euro will be dedicated to the actions that will be implemented through the missions, the soil and ocean missions. Another initiative that I think it's very interesting for you is the Circular Bio-Based Europe Partnership that will be launched very soon at the beginning of 2022, that will have a budget for the next years of also 1,000 million euro. So if we take into consideration this, you will see that for the actions that will be included in the work programs from 2021 until 2027, we will have around 6,500 million euros. So as I said, it's about research, but it's also about policies. And for researchers, probably it's difficult to come to a little bit higher level, to the policy level. When you start thinking about promoting a project, you cannot forget this political level, this that is defined within the strategic plan for the next 4 years, the strategic plan that will cover the period 2021-2024. And this document is completely linked to the definition of the work programs that will cover 2021, 2022, and the next period for the next 2 years. Because this is a strategic plan identifies the key strategic orientations on research and innovation, and also the expected impacts at political level. It identifies 32 expected impacts that are absolutely connected to the work programs and to the destinations, and of course to the topics that you will look into and you will compete in the proposals. Because here is the relationship. The relationship according to this Cluster 6 that we are presenting today. This strategic plan identifies 6 main impacts, which are the political priorities. In first place, climate neutrality and adaptation to climate change. The second one, the preservation and restoration of biodiversity and ecosystems. The third impact, which has to do with sustainable and circular management of natural resources, tackling pollution and bioeconomy. The fourth one, about food and nutrition security for all from sustainable food system from farm to fork. The fifth impact, looking forward balanced development of rural, coastal, and urban areas. And the last one, to develop innovative governance models enabling sustainability environmental observation. So if we look at these impacts, they are absolutely related to the destinations, to the several destinations that you will find in the work program, where the different research lines are included. And if we look at the calls that we have open now since the 28th of October, we have a very significant budget available, 861 million euros dedicated to the calls of these 7 destinations, several calls with several numbers of topics that I will describe very briefly now. And as you probably know, all this information is included in the work program that is open now and that covers the period 21 and 22. As you probably know, the topics for 2021 are already closed. They close on the 6th of October, but we have now new opportunities, those topics of the calls 2022. This is a very long document, 568 pages. It's similar to what we had in the past in Horizon 2020, but there are some new things that you have to take into consideration if you are thinking in participating on or coordinating an action here. And of course, as you know, if you apply for a topic, you have to cover and reply to all the requirements that is included in the calls. You have to take into consideration the specific conditions, the expected outcomes, the scopes, but also all those other words that are hidden in the topic, such as links to policies, links to specific, the requirements to include specific countries or regions of the world, those requirements regarding social sciences and humanities, gender issues, or other cross-cutting issues such as multi-actor approach. But there is something new in this Horizon Europe program, and is related to the impacts. This impact that is related to this strategic plan that I just mentioned at political level. All these expected impacts are described at destination level, and when you prepare a proposal, you also have to consider all these impacts that are described in this part of the work program. So please look carefully at this. These are all the calls that are included in this work program to 2021 to 2022. The ones of the first year, as I already mentioned, are already closed, and now we have opened several calls for 2022 that will be evaluated, in some of them in one stage and others on 2 stages. Please keep in mind these dates. As I said, the 28th of October, all of them were open. There is a first deadline, the 15th of February, for all of these calls, the ones in one stage, but also the first stage of the calls that are evaluated in 2 stages. For those proposals that go over and pass the first stage evaluation, you will have a second date in September. The calls on biodiversity and circular bioeconomy will have the second date on the 1st of September. And the 2 stages calls for farm to fork and communities will have another date for the closing of the second stage, that will be the 6th of September. As you know, all these projects are implemented using several types of actions. The most popular one are the research and innovation actions. In this case, out of the 77 topics that are open now within all these calls, 42 are carried out using RIAs, this research and innovation actions. 21 of the topics are implemented using innovation actions that, as you know, are closer to the market. And we also have 13 topics that will be implemented through coordination and support actions. And let's go through the different destinations, the 7 of them, very, very briefly, because we only have some minutes. I invite you to carefully look at all the content of the work program, particularly the index, where you will see all the calls and all the topics that are open now. The first destination is about biodiversity and ecosystem services, and for sure you will find, looking at your activities, you will find opportunities to carry out your research and innovation programs. Here, the European Commission will dedicate 141 million euro for 2 calls on single stage and 2 stages. And the purpose here is to answer to the challenges about understanding the biodiversity decline, A second area for valuing and restoring biodiversity and ecosystem services. The third area is about managing biodiversity in the primary production. Here is the bigger budget, about 78 million, will be dedicated to 6 research and lines. Then we have 2 topics about enabling transformative change on biodiversity, and 1 topic for interconnecting biodiversity research and supporting policies. Now I have some slides with all the details of all the calls and the topics. I will not go into detail because we would need more time, but following the indications from my colleagues from the University of Valencia, I have highlighted in red some of the topics that might be of interest for the audience today. Here you will find the title of the topic, the code for identification code for the call, for the topic. Also the type of action. Also you will see the European contribution, the budget that is suggested for each proposal, and therefore the number of projects that are expected to be supported and financed. Let me read the first one. Is the topic Biodiversity 0103. The purpose is to carry out a network for nature, a multi-stakeholder dialogue platform to promote nature-based solutions. This is a coordination and support actions of 6 million euros. And then we have in the block for interconnecting biodiversity research and supporting policies, you have the topic Biodiversity 0110, which is a coordination and support action that is looking forward the cooperation with the Convention on Biological Diversity. If we go to the second destination, fair, healthy, and environment-friendly food system from primary production to consumption, the purpose of this destination is to contribute to implement the strategy Farm to Fork that, as you know, is a pillar within the Green Deal strategy. This destination has a budget for this year 2022 of 231 million euros that will be dedicated to the following areas. In first place, near 80 million euros will be dedicated to enabling sustainable farming, 40 million to enabling sustainable fisheries and aquaculture, 57 million towards transforming food systems for health, sustainability, and inclusion. And there is an area for collaborating with Africa, 4 topics with a budget of 54 million euros. As you see here in this slide, the most common instrument to carry out research within this destination are research and innovation actions. You can see that 15 out of the 20 topics will be carried out through RIAs, these research and innovation actions. And let's go through some of the research lines that are open. You will see on a 2-stage evaluation process, you will find a topic which is the Farm to Fork 0202 that will deal with emerging and future risk to plant health. Here you have a research and innovation action with 7 million euro, and it is expected to support 1 project. Also, there is a research line on ecology of infectious animal diseases. Here you have research and innovation action. The budget is 12 million euros, so the European Commission will be able to support 2 projects with a budget of 6 million euro each of them. Let's go to the third area, which is about transforming food system for health, sustainability, and inclusion. Here you will find the topic Farm to Fork 0107, that is about building alternative protein-friendly, sustainable, and healthy food environments. This is an innovation action with a budget of 12 million euro to support one project. And then we have another line which might be very interesting for the audience today, which is the Farm to Fork 0109 with title Microbiomes in Food Production Systems. This is a research and innovation action with a budget of 10 million euro, and it is expected that each project will have a European contribution of 5 million. So it is expected that two projects will be supported in this line. And let's go to the third destination, which is a call for supporting circular economy and the bioeconomy sectors. This includes the traditional sectors such as textiles, plastics, construction, or electronics, but also those sectors linked to the bioeconomy, the forestry sector, or the blue economy. Here, there are 142 million euro that will be dedicated to two calls, single stage and two stages. And the purpose here is supporting and enabling a circular economy transition, innovating sustainable bio-based systems and the bioeconomy, safeguarding the multiple functions of European forest, and the last area for innovating for blue bioeconomy and biotechnology value chains. In this area, indeed, after hearing your presentations, you will find several opportunities, and I have just selected or highlighted some that indeed will be of an interest for you. First, you will see in the area of innovating for blue bioeconomy, the topic Circ Bio 0107 about marine microbiome for a healthy ocean and sustainable blue bioeconomy. This will be implemented through a research and innovation action. The budget available is 18 million euros, so it is expected that two projects of around 9 million euro will be financed. Then we have a big area with 74 million euros. All these research lines that you can see here, I have not selected any of them, but for sure you will see opportunity. I am looking here that there is one in two stages, Circ Bio 0202, which is about exploring extreme environments, novel adaptation strategies at molecular level for bio-based innovation. The last presentation, indeed, this topic is interesting for this research group. There is 10 million euro to implement research and innovation projects with a budget of 5 million euro. And I'm sure that indeed there are other opportunities within this block that I am not going into because we're lacking out of time. And now let's go to the fourth destination, which is about clean environment and zero pollution, where the main goal is to contribute to achieve this ambitious objective of the European Green Deal and its action plan towards zero pollution. You have now open calls with 51 million euro that will promote halting emissions of pollutants to soils and water. Here you will find three topics with 36 million euro. And the second area is about protecting drinking water and managing urban water pollution, one topic with 15 million euros. The destination number 5, 6, and 7, I will not go into the details of the topics because I think they are not so much related to the areas of research, so I will just go through them in general terms. Destination 5, it's about land, ocean, and water for life, for climate action. The purpose here is to study the impacts that climate change is having on land and marine environments, on natural resources, agriculture, forestry, and food systems. It's about studying the relation between ocean and climate also about the potential of ecosystems, the mitigation potential of ecosystems, the role of agriculture in order to reduce and mitigate greenhouse gas emissions, and also the resilience of agriculture and forestry to climate change. Destination number 6, with the calls about communities, is about investigating on resilient, inclusive, healthy, and green rural, coastal, and urban communities. This destination is about empowering the different actors of the different environments, the rural, the coastal, and the urban, and finding solutions so that the rural communities are the digital technologies give solutions to the problems that the rural areas are facing. Also the coastal communities and also the urban and peri-urban communities. The pandemic, the COVID-19, has shown us that the vulnerability of the urban community, if we think about supply of food, for example. All these research and innovation lines are included in this destination number 6. And the last one is destination number 7. It's about research on innovative governance, about using all the application about environmental observation and digital solutions in order to support the Green Deal challenges. As I said, well, such important challenges require new governance models that need the support of environmental observation, about the development and application of digital technologies. And also you will find here within this destination several research lines that they will support the agriculture knowledge and innovation system. This destination has a very important budget. About 147 million euro will be dedicated to 15 topics. And here you will see that due to the nature of this research, which is more about supporting policies, you will find a lot of topics that are carried out with coordination and support actions. Let me finish presenting the people that we are working with in my organization, which is CDTI, the Center for Development of Industrial Technology, within this cluster 6. And also, let me inform you about the website that our ministry has launched in order to inform about Horizon Europe, the events and the national contact points that are giving support to applicants. And you can see here our personal details, our telephone number and emails. So you can contact us in case you have any question regarding all the information that I have provided. And for Spanish applicants, in case they want to participate and promote the coordination of any actions, please contact us and we will do our best to support you and solve any questions you may have. We have organized several events at national level, but in first place, I would like to recommend you, if you are trying to participate or coordinate a proposal, please hear the recording and please see the presentations that the European Commission made during the 25th and 26th of October, because deep explanations about the content of the topics and recommendation on how to prepare successful proposals were provided during this info day. So I really encourage you to hear all this information. Beside that, for the Spanish applicants, I have checked the participant list within this webinar. I have seen that from the near 100 participants, around 9 or 10 participants were from Spanish organizations. I informed them that we'll be organizing we will be organizing a webinar to support coordinators on the 13th of January. This will be a virtual event. So in case you will coordinate a proposal, please contact us so we can support you until the deadline of the calls, which will be the 15th of February. I end up with some links to some documents that are indeed necessary to read and consult. The work program, the application forms, and some other manuals that the European Commission has developed. Please look at them carefully so that you will be able to prepare very good proposals. This is all from my side. Thank you very much for your attention. I hope that this information is useful, and don't hesitate to contact me in case you need further information. Thank you very much for your attention.
Thank you very much, Marta, for this really, really good presentation, a very good overview about the upcoming opportunities on Cluster 6. I'm pretty sure this was very useful indeed for many of our participants. Anyway, I open the floor to possible questions you might have. Marta, please.
I don't see any questions in the chat for now. Probably we can leave people to, we can give them a few minutes to find questions.
Yeah, I guess it's been a long day. We do have still 81 resistant and resilient participants with us at this point. It's very good to know that. Okay, well, I guess that since we'll be sharing all the presentations, you can actually find the contact details from everyone. So you will have the opportunity to contact people later. So I think we can move on. We are coming to the end of the webinar. So last call for questions. Well, if not, we can actually move on, if you agree. So just some final considerations from my side. If you allow me to ask you for a few more minutes. It's just an attempt to sum of this information up. I will share my screen again. Okay. Please tell me if you can see the slides.
Yes.
Okay, perfect. Thank you. So this is kind of an attempt to sum everything up. This is an overview of MIRRI Strategic Research and Innovation Agenda. So I believe that with everything that has been said by our speakers, I hope it was clear for all of you that MIRRI can indeed be a valuable partner for research and innovation projects in our several strategic areas. So one by one, very quickly, area number 1, research and pathogenic microorganisms and human or human-animal infectious diseases. We can offer expertise and resources covering several topics, including human and animal health, microbial pathogens, infectious diseases, emerging diseases and zoonoses, epidemic and pandemic prevention and response, and diagnostics. In strategic area 2, research and development of new biopharmaceutical and therapeutic solutions, we offer expertise and resources covering innovative medicine and biopharmaceuticals, antimicrobial resistance, antimicrobials, vaccines, phage therapies, microbiota and microbiome therapeutics. In strategic area 3, research and development of new, safe, healthy, and sustainable food and feed products, we cover the topics of food security, food safety, food processing and preservation, food microbiome, sustainable and nutritious foods and diets, and functional foods, pre, pro, and postbiotics. Strategic area 4, resources and methods for biological management of soils and crops. We cover the topics of soil microbiomes and healthy soils, plant microbe interactions, plant health and crop productivity, biocontrol and biopesticides, and biofertilizers. Strategic Area 5, we offer expertise in terms of resources and methods for biomonitoring and or bioremediation of soils and waters, covering the topics of environmental pollution, clean soils and clean water, biomonitoring and bioremediation, microbial pathogens, persistent organic pollutants and plastics and microplastics. Strategic Area 6, research and development of renewable bio-based chemicals, materials and bioenergy sources. We cover the topics of circular economy, climate change, renewable bio-based chemicals and materials and biomaterials, and sustainable clean energy and bioenergy and fossil biofuels. Last but not least, Strategic Area 7, rescuing and preserving microbial biodiversity. We cover the topics of endangered environments and biodiversity loss, composition and functioning of microbial communities, microbial bioprospection, cultivation, and preservation. I'm sorry for this, I have to read this. Just to make clear that we have be clear in transmitting our message. Please, you can find all this information in our website, MIRRI.org, as well as in its strategic agenda. In short, what we can offer to you as a researcher or a company, if you are willing to have a collaboration under a research and innovation project, we can offer to you a single point of access to 50+ world-class biorepositories, a broad catalog of 400,000 high-quality microbial resources and data, state-of-the-art facilities and technological platforms, access to cutting-edge services, techniques, and technologies, top-level scientific and technical expertise, good training opportunities, and finally, tailor-made, flexible, and cost-competitive, and even sometimes cost-free solutions. If you are indeed interested in collaborating with us, please do not hesitate in contacting us through access@mirri.org. Our access officer, Adriana Chiarelli, will be glad to answer to your questions, and we will always be interested in analyzing together with you the possibilities that we have to collaborate, not only under Horizon Europe, but possibly under other initiatives and projects. Last but not least, if this is the case, if you are interested in having your country organization joining MIRRI, if you are not already, please do contact us at info@mirri.org. We will also be glad to discuss with you all the pros of being, the advantages of being part of MIRRI as a country or as an individual collection. So this is it from my side. I believe this was, in spite of being a very long day, it was a very productive one. Let me stop sharing. So I hope you have found this webinar useful for you. We have always the contact channels open in case you want to get some more information from our side. We are always willing to talk with you about possible questions that you might have. I also want to take this opportunity to remind you that tomorrow we will be holding another webinar. Our colleague Jean-Luc Lucas will be hosting another very good group of speakers that will be talking about cutting-edge technologies for 2030 microbial culture collections. They will be discussing microbiota conservation and characterization, Malditov, and genomics. So also very important topics for discussion among the cultural collection communities and beyond. A final word, just to thank again to all the speakers and all the people that who have been involved in the organization of this webinar and also on today's webinar, and especially to all of you who have attended to the webinar. In the end of the day, you are the reason for us to exist. We are here to provide you the access to our resources, to our facilities. So please, again, do not hesitate in contacting us in case you are interested in Access MIRRI. Thank you very much again, and I hope to see you all tomorrow.
Goodbye. Thanks.
Bye. Oh, what of you?
Bye and thanks.
Bye. Thank you.